Hi everyone, so the goal of this video is to understand neuron function. Specifically, can you explain the molecular basis of an action potential? So just taking a step back from that learning target, we're going to be talking about the key molecules and the key ions that are inside a single neuron and what makes a neuron actually work. And this word action potential is just a fancy word to describe a signal that is being sent down a neuron so it can communicate with other parts of our body, whether it's other neurons in our brain, other neurons in our body, muscles, glands, whatever. All right, so this is the picture that we saw in the last lesson. And again, the key idea here is we're going to look at how does a signal get sent from the cell body or from one side of the neuron all the way down the axon and then goes to another neuron or to a muscle or to a gland. So the key is the function of a signal being sent down this axon. And so again, you have millions, actually billions, 100 billion neurons in your brain, and they make vast connections throughout your brain, and it's all about those connections and the signals being sent from one neuron to another. So let's look at how are those signals sent. So when you zoom in on a cell membrane, if you zoomed in on the neuron, you looked at just the cell membrane, and you might have seen a picture like this if you've taken biology, or you might remember it from our class in the beginning of the year when we talked about the cell and how things get in the cell and out of the cell and active transport and passive transport. In this case, these channels facilitate diffusion of different ions. So we'll talk about those ions and how they play a key role in the neuron. So we call this like a resting membrane potential. So at rest, what does the neuron look like? So like all cells, a neuron has positive charges and negative charges. It has sodium, it has potassium, it has water, it has all kinds of things kind of flowing inside and out of the cell and trying to stay in equilibrium. Now, the difference here is that there's more positive charges on the outside and more negative charges on the inside. And that leads to a very small charge, or what we call a very small voltage. We can actually measure it, and it's negative 70 millivolts. So a very small voltage. You might have heard of volts and voltage, like you might have like a 9-volt battery lying around. Here we're dealing with millivolts, so very small voltage. All right, and so how does this voltage occur? How do we get negative 70? Well, there's these things called leak channels that let sodium in and let potassium out. Sodium is going down its concentration gradient and it's going to go from outside to in. Potassium's going down its concentration, it's going from inside to out. Now the key difference here though is that sodium, represented with red here, there's not as many of these channels as there are potassium channels. So some sodium ions can enter the cell, but lots of potassium can leave the cell. And if you have more positives leaving than entering, you're going to end up with more positives on the outside and less positives on the inside. And that means overall this charge inside is going to be negative, and overall this charge on the outside is going to be positive. All right? They're just moving down their concentration gradient, but this difference in leak channels really leads to more negative charge on the inside of a neuron. All right, and then the other key thing here is to mention are these other channels called voltage-gated channels. They're voltage-gated, meaning that they will respond to different voltages that are happening in the neuron. All right. Now, so far we've only talked about the neuron at rest, but now we're going to start talking about what happens with a neuron's action potential. When does it become active? So there's these voltage-gated channels, and they are going to open and close in response to different voltages. So let's say that there's some kind of stimulus, some kind of message gets sent to a neuron, and if that stimulus is big enough, then it's going to lead to an action potential. So when I say stimulus, that could be some other signal being sent from a neuron, and what actually happens with that stimulus is it causes some sodium ions to enter the cell. All right, and so as sodium ions enter the cell, that means positive charges are going in. And that means that our negative 70 is going to slowly creep up to something around negative 55. Now, and if you get to negative 55, that's what we call the activation threshold. That means that you've reached a critical point where now you're actually going to have an action potential and your neuron is going to fire and it's going to send a signal all the way down the axon. All right. And this whole process, or this first step, is called depolarization. The neuron is becoming depolarized. It's going from a negative 70 up to a number that's going to be closer to 0 is actually going to go past 0 and become even a little bit positive. So it's becoming depolarized. And again, sodium channels are going to open up, and that lets positive charges in, raising the voltage. All right, step 2. So we've reached a point where a lot of sodium ions have entered the cell. We've gone up to 0, we've even passed 0, and now the voltage inside the neuron is positive 30. There's so much sodium inside the neuron right now. So once you reach about 30 millivolts, a couple of things change inside the neuron. 1, the sodium channels close. 2, the potassium channels open up. All right, so there's these other voltage-gated channels for potassium. They're going to now open, and now potassium can start to leave the cell very easily. And so we have potassium leaving, and that's going to drive the voltage to the negative side again. And again, this process is called repolarization. So we're going to go back towards our negative 70, which was our resting number. So we're heading back towards the negative side now. Step 3 is sometimes called hyperpolarization, because not only does the neuron go back to negative 70, it actually overshoots a little bit, and it can end up going to somewhere near negative 90. So very quickly, a lot of potassium leaves, and you kind of overshoot your kind of goal of negative 70. It goes all the way to negative 90. So that's why it's called hyperpolarization. And at this point, both voltage gates close, and we're going to start to get back down to our normal levels. So the voltage gates close, and the neuron will soon restabilize at negative 70 millivolts. And once it gets back to negative 70 millivolts, then it can be ready to fire again. So you might hear these terms about a neuron firing. That is just this idea of an action potential being sent down the axon. And once everything kind of gets reset, then the neuron can be fired again. So you can think about your muscles, and maybe you're working out and you're telling your muscles to work, you're telling your biceps to work, and those signals are being sent to your muscles, and your axons have to consistently fire, and then if you're telling your muscle to keep working, that signal keeps on firing and firing and firing. And so the axon needs a little bit of time to kind of get back to normal. But if you're still sending that signal with your brain, the axons are just going to fire and fire and fire and fire. As soon as they get reset to negative 70, they're going to fire again. And so what you might see every now and then is you might see a graph that kind of looks like this. One thing that I think is very interesting, one thing I'll point out, is just the time scale. We're dealing with milliseconds. So we kind of talked about like reaction time and how that can be on like a few hundred milliseconds. When it comes to an individual neuron, everything here is happening within a few milliseconds. So I'm talking about some kind of stimulus happens. If that stimulus is big enough, if it reaches the threshold, then we're going to go down this path of depolarization, where sodium ions enter, and then repolarization, where potassium ions leave. We overshoot our goal of negative 70, we get our hyperpolarization, and then things kind of reset and we get back to our resting state. Now, like I said before, this is where a neuron can actually rest, or if you're telling it to do a job again, it's going to kind of jump back to this step here where now it's at negative 70, we can have another stimulus and it can do its job one more time. But this all happens in a couple of milliseconds. All right, so I think that's where we'll stop here, and that is how neurons work. So good luck with your notes and good luck with answering any questions about neuron function.